Assalamualaikum and hi everyone. In this video, my group will explain about chromatography in pharmaceutical, which is drug taste in urine. So let's start with the introduction. What is chromatography? Chromatography is a laboratory technique for the separation of a mixture into its components. The mixture is dissolved in a fluid solvent. It can be a gas or liquid called the mobile phase, which carries it through a system such as a column, a capillary tube, a plate, or a sheet, of which a material called the stationary phase is fixed. The separation is based on the differential partitioning between the mobile and the stationary phases. So in this video, we will use the application of paper chromatography to test the drug in urine. So what is paper chromatography? It is a technique that uses paper sheet or strip as the absorbent being the stationary phase through which a solution is made to pass. The mobile phase is a solution that travels up to the stationary phase due to the capillary action. The mobile phase is generally a mixture of non-polar organic solvent, while the stationary phase is a polar inorganic solvent such as water. Principle of paper chromatography is the substance are distributed between liquid phases. The two phases are water held in pores of the filter paper, and the other phase is a mobile phase which passes through the paper. When the mobile phase moves, the separation of the mixture takes place. The compounds in the mixture separate themselves based on the difference in their affinity towards stationary and mobile phase solvents under the capillary action of pores in the paper.
Hi, Assalamualaikum. My name is Mira Ardila binti Mat Johari. I am going to tell you the objective of the experiment chromatography in pharmaceutical drug test in urine. The first one is to separate the athlete's urine compound, and the second one is to detect the presence of drug in athlete's urine. Next, I will be presenting about the theory of paper chromatography. Paper chromatography is an analytical method that is capable of precisely determining compounds in the bloodstream. This technique uses biological samples like urine for testing in order to identify drugs and other doping agents that are frequently used in sports. Both polar and non-polar solutes can be separated using paper chromatography. Drug-infused urine usually has a higher polarity than regular urine.
Assalamualaikum warahmatullahi wabarakatuh. My name is Ko Ani Sofia binti Ku Muhammad Zaki Amani, and now I will be explaining about the methodology for our project. These are the apparatuses and chemical reagents that we will be using. Moving on to the procedure, we will first look at the preparation for the samples. Firstly, we will need to obtain the urine samples from 3 different athletes, and then 3 5 ml beakers are prepared and we label them as A, B, and C. Then, transfer the samples separately into 3 beakers. Then, we will look at the preparation for developing solvent. To prepare the developing solvent, we mix 10 ml of petroleum ether, 1 ml of isopropanol, and half a drop of distilled water into a beaker. The developing solvent is then poured into a 150 ml beaker to the depth of approximately 0.5 cm. After that, we close the beaker and shake it slowly to ensure that they are in an equilibrium state. Then lastly, we cover the beaker properly. Now, we will look at how we apply the samples to the TLC plate. Firstly, by using a pencil, a line will be drawn at the bottom of the plate. Then, mark 3 small X's along the line. We make sure that they are distanced equally. And then label A, B, and C at the bottom of each of the X's. By using a capillary tube, we place a tiny spot of each of the urine samples on each X mark. And the last part of our methodology is developing the TLC plate. To develop the TLC plate, we place it carefully into 150 ml beaker that contains the developing solvent. The level of the solvent is below the line drawn. And when the solvent has reached 3/4 of the length of the plate, we mark the position of the solvent front and take the plate out of the beaker. Lastly, we visualize the spots, measure the length of the spots, and calculate the RF for each of the sample. For further discussion about the samples, we will now continue with the results.
Next, we will move on to the result. The thin layer chromatography plate shown that A. Testosterone less polar, B. Normal urine, C. Amphetamine more polar. Compounds that are more polar will wander farther on the plate because they experience less attraction to the stationary material, while compounds that are more polar will wander lower on the plate because they experience more attraction to the stationary material. RF value calculation. RF value is a measurement of how far the solvent liquid that the compound was dissolved. So we will be using a formula RF equal to distance solid move divided by distance solvent front move. Given that the solvent front is 10 cm, so the calculated RF value is RF value for A equal to 9 cm divided by 10 cm equal to 0.9. RF value for C is 8.5 cm divided by 10 cm equal to 0.85. Polarity. Therefore, this is proven that testosterone is less polar than amphetamine because its net dipole moment equal to 0. Meanwhile, amphetamine is more polar because its net dipole moment is not equal to 0. Also, chromatography shown that A travels slightly farther than B, which we can differentiate its polarity from it. Less polar travel farther distance than more polar.
Hi, my name is Maizarul Hezikri and my metric number is 20218934 and 32. And today I will present about the advantages of using chromatography technique in determining the drug in urine. Firstly, the advantages is the component of complex mixture is easily separate and recover. And then the non-volatile compound can be separated by this method. Next, all component of UV light is achievable to visualize. And lastly, the separation is done in a very short time as the component elutes rapidly. For the conclusion, liquid chromatography is a useful analytical tool for establishing the component of a drug formulation, enabling researchers to quantify the formulation and discover whether there are any impurities in a product. Next, based on the drug test in urine that we have done, the urine sample labeled as B have no drugs, while the urine sample A and C contain drugs. And lastly, this is our group reference for the drug test in urine experiment. Thank you and Assalamualaikum.